vacuum of space, astronauts are exposed to cosmic and solar radiation constantly. This can damage cells and surrounding tissues, leading to mutations and inflammation. Such systemic responses are precursors to cancer, vision impairment, or disruption of the central nervous system, just to cite a few known effects. This is a limiting factor in our ambitions to establish a permanent base on the moon and one day, hopefully not too far away, send human explorers to the surface of Mars. While the link between radiation and cancers has long been understood, machine learning has the potential to help us understand the specific molecular drivers of cellular damage in space radiation exposure, and do it in a way which is private and secure, the way all patient data should be. I am therefore pleased to be able to introduce the FDL 21 astronaut team.
Hello everyone, it's my pleasure to introduce the Astronaut Health team. My name is Owen and I'm an MD PhD student studying health data science at Oxford. Over the last 8 weeks, our work is focused on bringing together 3 areas of research: causal inference, federated learning, and domain generalization. I'll explain what each of these mean in the presentation. But first, I'll introduce the team. I'm one of the 4 researchers on this year's team, and my fellow researchers are Paul, Giuseppe, and Linus. Supporting us have been our amazing faculty from NASA, Intel, and the Mayo Clinic, last year's Astronaut Health team, and last but certainly not least, our team leads, Lauren Sanders and Frank Sobachensky. Our work this summer is motivated by the biological challenge of spaceflight, and specifically cosmic radiation. This radiation can penetrate through thick layers of shielding and body tissue, and on a 7-month trip to Mars, it's cause for concern. Cosmic radiation is carcinogenic. And if we are ever to succeed as a spacefaring civilization, this is an issue we must address. Understanding the exact mechanisms of radiation damage can give us huge insight into how to reduce and prevent damage. However, studying these mechanisms is difficult. This is as human space data is incredibly expensive to generate. The data we have is also incredibly siloed in separate locations. Furthermore, when data is millions of miles away in space, getting it all back to Earth is hard. There is, however, another data source, which are animal models. While this data is not exactly human, we can generate large volumes of samples without these siloed data issues. This brings us to our research question. Can we combine high-dimensional human and mouse data to learn about human radiation risks using data stored in different locations? In this question, I firstly want to highlight the idea of combining human and mouse data. This has been of particular interest in NASA, who want to make the most of the data they have in a combined setting. I secondly want to highlight analyzing data stored in different locations. This has been of particular interest to our partners at Intel. I'll now hand over to Paul.
Thank you, Owen. I'm Paul Duckworth, a postdoctoral researcher at the Oxford Robotics Institute at the University of Oxford. As Owen mentioned, our project built upon last year's Astronaut Health team and enhanced the causal inference platform, CRISP, in various new ways, given new priorities from our partners. In total, we expanded upon the work in 3 directions. First of all, we expanded the causal machine learning methods in CRISP to cover a wider range of problems and data types. Secondly, to tackle the problem of siloed data sets, we developed a proof of concept pipeline to learn an ensemble of causal models without the need to move data between physical locations. Finally, using radiation exposure data from mice and humans, we investigated transfer learning between organisms to overcome the problem of small data sets in biological settings. We all know that correlation does not imply causation, and we wish to learn the causal features that can be used to predict even on out-of-distribution test cases or under distributional shift. We wish to identify the features of our dataset that are consistently predictive of a target across many different environments, such as in the example image shown here. These invariant features are then considered candidates for being causal features. Medical data is often siloed in different locations, so CRISP version 2.0 implements causal methods in a federated framework. Here is an overview of the federation architecture based on Intel's open source software, OpenFL. Starting on the left, we have a global model that can be used for pre-training or initializing parameters. This model is then shared with a set of collaborator institutions or users who each use the model and train one round of training on their local data sets. The models are then updated by the users and sent back to the aggregator to average the model parameters and reshare the new model with the users. All institutes train each model in the ensemble on their data sets, and no data is moved during the process. In summary, CRISP version 2.0 pipeline can be realized in either a federated or non-federated setting. It begins its life on GitLab, and it is fully Dockerized and outputs an ensemble of results to a front-end Streamlit application for further analysis and insights. I'll now hand over to Giuseppe to discuss our data sets.
Thank you, Paul. I'm Giuseppe from the Maths Department at Oxford. I will start by showing you how models trained in a federated way match those trained in a non-federated way. We validate this on synthetic data sets, which allows us to consider different known causal structures. In the shown plot, the y-axis corresponds to the expected ratio of causal features found when considering alpha elements. The higher a plot is above the random sampling line, the better. As shown, the federated implementations in the orange line perform as well as in the non-federated case, in the blue line. Now that we have validated the accuracy of federated methods, we can shift our focus back to the aim of the project, inferring causality relations for carcinogenesis with human and mouse data. Despite the differences in the biological background of the human and mouse data, we hypothesize that there might be a benefit to the performance of our causal inference methods by mixing them. Therefore, we make the human and mouse data coherent by identifying genes with similar structures and function between the two organisms. To that end, we utilize the ensemble database maintained by the European Molecular Biology Laboratory. I will now hand it over to Linus, who is going to walk you through the results of the biological data.
Thank you, Giuseppe. I'm Linus and I'm a PhD student in computer science at the University of St. Gallen in Switzerland. I would like to highlight some of the most striking biological results that we obtained with CRISP 2.0. To overcome the issues of small data set size and to improve generalization capabilities, we ran a set of experiments to understand how our models performed when using varying degrees of human and mouse data. Using all available human transcriptomic radiation exposure data, we progressively added mouse data until the mice outnumbered the human sample points 2 to 1. What we find is that our results are strikingly consistent at different levels of blended data. The figures on the right show how much concordance there is compared to 100% human data in the top 10 and top 20 features that our models predict. These results suggest that for experiments studying the effect of gamma radiation on blood, it is possible to blend human and mouse data in causal inference settings to still identify human relevant features. Therefore, I would now like to showcase one result obtained on this blended gamma radiation exposure data. We used linear IRM, a causal inference method that is part of CRISP, to identify genes with invariant relationships across environments to predict the radiation state. The absolute coefficient score, seen in the blue bars, corresponds to how certain the model was of an invariant and causal relationship to the biological effects of radiation exposure. Checks with the literature reveal that our models function as intended. The identified top factors are strongly linked to cancer in published articles. Of particular interest is that we could not find published research on a top scoring gene. This suggests that there may be value in studying this compound in more depth. In summary, the FDL Astronaut Health project this year showcased the utility of causal and federated learning on biological data to advance our understanding of space radiation risks. We investigated approaches like the merging of human mouse data to deal with low sample sizes, and also performed comprehensive validation on synthetic data sets. On top of that, we also presented a working prototype of a federated ensemble of causal machine learning methods. There are still some next steps that we aim to tackle in the future. These include new approaches for causal feature weighting, the inclusion of more causal discovery methods, as well as applying a federated workflow on real-world distributed datasets. Thank you very much for your attention.
Amazing. Thank you so much, Astronaut Health. That was fantastic, and you set the bar very high. So now I'm going to go ahead and turn it to Bill to start our Y panel and introduce our panelist.
Great, thank you. Well, again, to echo Sarah, that's a wonderful, wonderful job, but an amazing continuation of the work done last year in the development of the CRISP tool. And I think the implications here are amazing. To share this little review with me, we have Scott Penberthy, who is the Chief Technology Officer at Google Cloud, and he joins us today from Lake Tahoe in sunny and smoky California. And also with us is Shashi Jain. He is the Tech Innovation Lead for Health Solutions at Intel, and I believe he's joining us from up in Portland. So Scott, starting with you, what are the implications here in terms of real-world applications and improvements to things like early diagnostics in medicine with the work that this team has done in the area of causal inference?
Well, I think it's this is where it's at in medicine, Bill. And I think we're seeing now that we're talking a lot about precision outcomes. It's producing a fountain of Nobel laureates and national medal winners these days. And I'm just curious, maybe for the team itself, because I've been following some of the work. It's been like Chris Mason and whatnot, and looking at tardigrades and how they have these suppressors, the D sub protein, if you will. I was curious, if you look at what's happening in the humans and the mice, are there things that are helping repair our DNA that for like what happens for sun radiation and whatnot? And are you starting to find those genes in in your analysis? And then the second part is, I love this statement of it's correlation is not causality. And it's very interesting, and I like your insight to say what's invariant between the environments. Maybe those are things that actually lead to causal inference. So that's a brilliant insight. So I think there's no shortage of people who are very interested in your outcome, but I think just your opinion of like, what are you finding versus the work around DSWA, if you're familiar with that, and how might those two relate is my question.
Wonderful. And Shashi, can you tell us a little bit more about Intel's federated learning technology, and which was clearly a big part of this work? Can you describe what this is and what your vision is for federated learning?
Yeah, certainly. So federated learning, again, is a technology where you can train an algorithm on data that's siloed or heavily protected, or that needs to be kept private. And astronaut data absolutely fits the bill. There's only a few astronauts that have gone into space, and their data is highly protected. Similarly, mouse data from the Mayo Clinic, and there's many other sources. So what federated learning allows is for the algorithm to go to the data versus the data being aggregated together to train the algorithm and then improve it over time. So the federated learning software is this is an open source project. You can check it out on GitHub if you'd like. But the vision here is to enable many more collaborators and increase the pace of innovation where there are these data silos. And what I'm excited about in this in the approach that this team took is they not only proved that a federation could be set up very quickly. In fact, they set it up probably quicker than I did on my own bench. It was great to see that, but that they could increase the number of collaborators that have. Admittedly, there's going to be very few that have astronaut data with radiation testing, but now they've opened up to different kinds of data. Collaborators that have radiation testing data elsewhere, and then they've developed a system that can increase the pace of innovation. So the operations side of this, I think, is just as important as the research side here.
Yeah, absolutely. And by the way, Shashi, I want to point out for all our listeners that we do ask all the mice to read and acknowledge our privacy policy where their data is concerned. So it's not just about the astronauts. Scott, a closing comment here. What for you is a key takeaway from this team's work and what they presented to us today?
Wow. It's just wow. It's such brilliance, and what you did in 8 weeks, it's not worthy. That's a heck of an accomplishment in the period of time. And I think you are on the cusp of what's happening in research in medicine. And I look forward, maybe one of you or all of you will at some point crack into the Nobel Prize. So I'm really interested to see where you take this.
Wonderful. Thanks.